Hello, I'm Yong Gu, a former postdoctoral researcher in Dr. Mark Ruella lab. Today, on behalf of Dr. Mark Ruella lab, I'm going to introduce interesting study where Dr. Ruella conducted to understand the resistant mechanism of the CAR T-cell therapy. As many of you know well, the chimeric antigen receptor T-cell therapy is a new form of immunotherapy that includes the genetic engineering of patient T-cell to redirect them against the cancer. Anti-CD19 CAR T-cell have been particularly successful against the B-cell leukemia and lymphoma, with the first CART19 product to be approved by the FDA being developed at UPENN. Now we have multiple FDA-approved CAR T-cell, as shown here, for various indications, including B-cell leukemia, lymphoma, and multiple myeloma. As mentioned earlier, the CAR T-cell therapy have achieved great success in targeting the B-cell leukemia and lymphoma. Briefly, the 80% of leukemia patients showed a complete remission rate, and around 50% of lymphoma patients have achieved a complete remission rate after CART19 treatment. Despite this great anti-tumor efficacy, not all patients have same experience. As shown here, the more than 50% of leukemia and lymphoma patients still show no response or some response initially, but eventually tumor is relapsed. These clinical results strongly indicate that it is essential to develop a novel effective approach that can improve the CAR T-cells anti-tumor efficacy, so we could help patients who currently have no other clinical options. But in order to develop the most potent CAR T-cell therapy, the first thing that we need to do is to understand the resistant mechanism that drive the failure of the CAR T-cells therapy. As you may know, there are multiple factors that can affect the CAR T-cells anti-tumor activity. For example, immunosuppressive tumor microenvironment, including MDSC, TOM, Treg, could reduce the CAR T-cells anti-tumor activity by secreting the various immunosuppressive cytokines. In addition to these extrinsic factors, tumor itself could also generate the multiple resistant mechanism to CAR T-cell therapy by modulating their molecular features, such as the expression level of the inhibitory receptor on their surface. Despite multiple studies found the important biological factors that can reduce the CAR T-cells anti-tumor activity, it may be quite difficult to identify the most important molecular features in cancer that dominantly lead to the failure of the CAR T-cell therapy, since the design of most previous study was mainly focused on the specific the molecular pathway based on their interest. So to overcome this problem, Dr. Nathan Singh in Dr. Mark Ruella lab performed a very interesting research. In this study, they hypothesized that the most important tumor intrinsic factor that can affect the CAR T-cells anti-tumor activity can be identified by functional genomics approach using the genome-wide CRISPR knockout screening platform. Before introducing the data, let me briefly go over the protocol of the study. In this genome-wide CRISPR knockout screening, they used a pool of lentiviral CRISPR libraries named Brunello libraries that target the 20,000 different genes by 76,000 different single guide RNA. With this lentiviral vector containing of each guide RNA and Cas9, they generate the lentivirus, and then they transduce this lentivirus into the B-cell leukemia cells called NAM6, and they select the transduced cell by using the pyromycin. After selecting the transduced NAM6, they co-culture them with the CART19 and harvest the survived NAM6 to identify what tumor intrinsic factor affect the CAR T cells anti-tumor activity by performing the next generation sequencing. With this protocol, they identified various interesting results, and today I'm going to introduce some key results. First, they found that the NAM6 with the knockout of positive regulators of extrinsic apoptosis such as caspase-8, FAD, and BEAD, and DAS receptor 5 survived much better in the presence of the CART19 than the one that had the knockout of the negative regulator of extrinsic apoptosis, including BERK, CFLIP, TRAF2. In addition, the gene set enrichment analysis also demonstrate that the apoptosis was the most enriched pathway, and these results strongly suggest that the modulation of apoptosis, especially in DAS receptor mediated extrinsic apoptosis in cancer, may have substantial impact on the sensitivity of cancer cell to CAR T cell mediated killing. For those who are not familiar with apoptosis, let me briefly introduce the general idea of the apoptosis. Apoptosis can be classified into two pathways, such as intrinsic pathway and extrinsic apoptotic pathway. First, the intrinsic apoptotic pathway can be triggered by factors like chemotherapy, radiation, DNA damage. And this intrinsic apoptosis is strongly associated with mitochondrial-mediated apoptosis. For instance, when the intrinsic apoptotic pathway is triggered, the BH3 omniprotein like the BAM, BEAD, BAD inhibit the function of the BCL2, which is the key anti-apoptotic regulator of intrinsic apoptotic pathway. Upon inhibition of BCL2, BACs form homodimer and destabilize the membrane of mitochondria. As a result, pro-apoptotic molecules such as cytochrome C is released and initiate apoptosis. On the other hand, extrinsic apoptosis can be triggered when DAS receptor ligand, such as FAST ligand, TNF-alpha, TRAIL, interact with their associated receptor like FAST, TNF-alpha receptor, and TRAIL receptor. Upon binding of the DAS ligands to their associated receptor, the pro-apoptotic regulator like a FAD and caspase-8 are formed, DAS-inducing signaling complex, to promote the downstream of the apoptotic signal pathway. However, to inhibit this extrinsic apoptotic pathway, anti-apoptotic molecules such as FLIP, CIAP, and TRAF2 plays an important role, as I introduced in previous study. And as I introduced in previous study, the Nathan found that the cancer cell become more resistant to CAR T cell therapy when caspase 8 were, when FAD and caspase-8 were knocked out. On the other hand, the lack of anti-apoptotic molecules such as the CIAP, CFLIP, and TRAF2 induced the CAR T cell mediated tumor killing. So to validate their genome-wide CRISPR screening, Nathan generate the FAD and BEAD knockout, B-cell leukemia cells, and study whether this apoptotic resistant cancer cell can reduce the CAR T cells anti-tumor activity. As shown here, the FAD and the BEAD knockout dramatically reduced the CAR T cells anti-tumor activity in vitro and in vivo, suggesting that the modulation of apoptosis sensitivity in cancer cell can affect the CAR T cells anti-tumor activity. To identify the molecular programs responsible for the CAR-driven T cell dysfunction, they performed the transcriptomic and epigenomic investigation of the CAR T cell exposed to wild type and BEAD knockout NAM6. In the analysis of the gene expression profiles, of the CAR T cell at rest or after 15 days of the chronic exposure to wild type or beat knockout NAM6 cells, they identified around 1,200 differentially expressed genes. More specifically, CAR T cells exposed to the beat knockout NAM6 cells demonstrate increased expression of the known dysfunction associated genes, such as the EGR2, CTLA4, and IRF4, BTLA, TOX2, and NR4A2. In addition, they also identified the decreased expression of the T-cell activating genes, including T-cell signaling molecules such as SLAMF6, LCK, JAK1, and SOS1. And moreover, stimulatory surface receptor and transcriptional activators. In addition, to study the CAR-T, they also study the CART19 chromatin accessibility to determine if persistence and antigen exposure result in epigenetic imprinting associated with the dysfunction. Similar with the RNA sequencing data, they found that they enhanced the promoter accessibility of several key immunosuppressive transcription factors, such as TOX2, IRF8, and PRDM1 in beat knockout exposed to CART19. So together, these findings suggest that the persistent exposure to apoptosis-resistant leukemia lead to the global programmatic change in CART19 cells that are similar with the T-cell exhaustion, which eventually lead to the failure of the CAR T cells therapeutic effect. Finally, they confirmed their preclinical result by evaluating the clinical samples. To this end, they evaluate the expression of a group of genes that plays the key role in inducing DAS receptor-mediated apoptosis in the leukemia cells from the ALL patient who treat with the CART19. Upon the expression level of the positive regulators in DAS receptor-mediated apoptosis, they then divide the patient into two different groups. First, high score group, where the patient have a higher expression level of the positive regulators of DAS receptor-mediated apoptosis. Second, low score group, which have the lower expression of these positive regulators. And very interestingly, as shown here, what they found that the patient with a high score, meaning the high level of positive regulator of DAS receptor, showed a significant higher overall survival rate as compared to the lower group. So this data clearly support that the sensitivity of apoptosis, particularly DAS receptor-mediated apoptosis in cancer, have substantial impact on the CAR T cells anti-tumor activity. So in addition to the tumor intrinsic factors that cause the failure of the CAR T cell therapy, Dr. Ruella also found very interesting CAR T intrinsic factors that significantly impact on the CAR T cells anti-tumor activity. So as a second part of the talk, I'm going to introduce another study where Dr. Ruella studied how the CAR T cell signaling driven by the CAR T cell structure affect the CAR T cells anti-tumor activity. While Dr. Ruella group conducted two pilot clinical trials to test anti-tumor efficacy of the CD22 targeting CAR T cell in patients with relapsed or refractory leukemia, they observed unexpectedly low response rate in the trials as compared to the previous clinical outcome of the CD22 targeting CAR T cell developed by the NCI. While the direct comparison of distinct clinical trials at different institutions is inherently limited, differing the outcomes of these two trials of CART22 between NCI and the UPENN were striking. Basically, the PENN clinical outcome, in the PENN clinical outcome, Dr. Ruella observed a 50% complete remission rate, where the NCI trials, they achieved a 73% CR rate. And these different clinical results actually led to explore the potential factor that may affect the overall CART22 anti-tumor efficacy. To understand the difference, they investigate multiple factors. For instance, the patient characteristic and manufacturing process in these trials were quite similar, and both CART constructs were composed of the M9071, which is the clone name of the CART22 SFB, as well as the same hinge and transmembrane and intracellular signaling domains. They eventually identified that these two constructs used in PENN and the NCI differed only in the length of the scFv linker as shown here. The CD22 CAR evaluated by NCI connected a heavy to light chain using a GGGS amino acid linker, just the one GGGS linker, while the trials of the University of Pennsylvania connected the variable chains using the 4 times lipid of the GGGS. So, which means that it has a much longer linker between the heavy to light chain. Therefore, based on this difference, they hypothesized that the scFv linker length may influence CAR T cells anti-tumor activity. So to validate their hypothesis, they first generate the CAR T cell with either a short or long scFv linker and perform the head-to-head comparison in the preclinical setting to see whether the difference of the linker length actually impact the overall CAR T cells anti-tumor efficacy, as we saw in the clinical outcome. So as shown here, the prolonged in vitro co-culture revealed the superior cytotoxicity of the CART22 short T cells. They contain the much short linker as compared to the one with the longer linker. And collection of the co-culture supernatant was also demonstrated that the higher secretion of pro-inflammatory cytokines such as interferon gamma, IL-2, and TNF-alpha by the CART22 short T cell, which is labeled as the green. So next, they also evaluate the in vivo activity of the CART22, which contains either short or long linker in the immunodeficient NSG mice bearing the B-cell acute lymphoblastic leukemia. So CART22 short T cell, as shown here, demonstrates a significant enhanced disease control, while the CART22 with the longer linker only partially reduced the disease burden compared to the control T cell. These differences were also reflected by the in vivo T cell expansion, as you can see here, and directly translate to the enhance the animal survival. So this in vitro and in vivo data confirm that the short linker could dramatically affect the CAR T cells anti-tumor activity. So to understand the molecular mechanism how the scFv linker length influence the CAR T cells anti-tumor activity, they first performed the biochemical evaluation. Measurement of the scFv, as shown here, revealed no significant difference between short and long linkers. Interestingly, predictive protein structure, as shown here, modeling of two SCFP suggested that the longer linker allowed the appropriate pairing of the heavy and light chain from the same molecules, while the short linker inhibit the intramolecular heavy and light chain pairing. This modeling is consistent with the previous SFP study demonstrating that linkers more than 9 amino acid in length allowed appropriate SFP folding. However, the linkers shorter than 9 amino acid in length are known to induce the rigid SFP structure that prohibit the intramolecular the variable chain pairing, and instead promote the intermolecular heavy and light chain pairing, which often result in the small increase in measured binding affinity as shown, as we can see in our construct. Evaluation of the SFP oligomerization using the size exclusion chromatography confirmed that the CART22 long exists as a monomer, while the CART22 short homodimerized. To better understand how SFP dimerization impact activity of the CAR molecules, we engineered T cell to express the GFP as shown here in a frame with a CAR polypeptide, allowing us to visualize the receptor distribution on the cell surface using confocal microscopy. CART22 long distribute uniformly across the cell surface as shown here, while the CART22 short appear to self-aggregate and cluster. And based on this observation, we hypothesized that the short linker promote the clustering of CAR, leading to the antigen-independent T cell activation. And this antigen-independent T cell activation eventually results in enhancement of the CAR T cells anti-tumor activity by priming the CAR T cell to activation. To understand the impact of the CAR clustering on antigen-independent receptor activity, they evaluate the difference in signaling protein activation driven by long and short receptor in the absence of target antigen by using the phosphopeptide array 28. The CAR clustering resulted in the greater phosphorylation of critical T cell signaling molecules in PI3K and MEP kinase pathways. They also observed increased activation of PPC3, a key cell cycle regulators protein. They also next investigate the transcriptional impact of this observed difference in tonic signaling. The gene set enrichment analysis revealed a significant increase in expression of cell cycle the programs. So together with the previous data, this data strongly suggests that the CAR clustering resulted in antigen-independent T cell activation, leading to increase of the CAR T cells anti-tumor efficacy. So in conclusion, Dr. Mark Ruella identified that the acquired resistance to apoptosis in cancer is a key feature that could contribute to the failure of the CAR T cell therapy. And unbiased genome-wide loss of function screening, they identified the critical importance of the death receptor signaling in tumor cells on CAR T cell cytotoxic functions. In addition, they also identified the shortening the amino acid linker connecting variable heavy and light chains of CAR antigen binding domain, drive the receptor homodimerization and antigen-independent signaling. And in contrast to the CD28-based CAR, the autonomously signaling 4-1BB-based CARs demonstrate enhanced immune synapse formation and activation of pro-inflammatory genes and superior effector function. Here are my acknowledgements. I thank all who were involved in these great studies, and especially I really appreciate Dr. Mark Ruella and Dr. Jun and Dr. Gill for their kind support. In addition, I would like to thank my foreign source which support my research. And thank you for your attention.